In the network analysis module, you can construct putative mechanisms that explain the changes measured in your experiment. You can get answers to complex biological questions, such as, what is the mechanism through which my treatment triggers apoptosis? Or, what is the mechanism through which this chemical affects cell cycle? In steps 1 and 2, iPathwayGuide allows you to select a precise set of genes of interest. In step 3, you may add gene ontology terms and chemicals to explore the mechanisms through which they are connected to your genes of interest. By default, all differentially expressed genes from your experiment are included in the genes of interest. However, you may wish to narrow your focus to a smaller set of genes. In step 1, iPathwayGuide allows you to select differentially expressed genes related to significant pathways, chemicals, or other analysis results. You may do this from each results panel using either the table or the dendrogram view. No matter how you make your selections, the resultant gene set will contain only the differentially expressed genes annotated to all of your selections. If 2 selections have no differentially expressed genes in common, your set of genes of interest will be empty. You may also select genes of interest in step 2, fine-tuning. From this section, you may remove genes you know to be irrelevant or add specific genes to your set, even if they are not differentially expressed or even measured in your experiment. Notice that as you select analysis results and fine-tune individual genes, your selections are recorded above the gene bar plot, and the bar plot itself is updated with all selected genes. These are the genes that will be included in your network analysis. If you ever want to start over, either with all differentially expressed genes or no genes at all, use the menu options above the gene bar plot. In step 3, you ask your biological question. For instance, if you would like to explore the mechanism through which a given drug or chemical triggers a given biological process, here is where you specify the chemical and the biological process. To select one, go to step 3 and choose the appropriate category for the type of mechanism you want to explore. Then use the search bar to find your process, function, component, or chemical by name. Add as many as you want from any category. When you are satisfied with your selection of genes, drugs, and Go terms, click Build Network to visualize how they are all interconnected.